Well, hello, geneticists. Pretty important day, our last inflection point into the third and final unit of the course, where we begin to talk about human genetic disorders. So, pretty exciting. This is the first of the 3 lectures on human genetic disorders, and we will first be talking about recessive traits in general. We'll be reviewing the genetics and molecular genetics of recessive traits. That'll lead us to considering recessive diseases more generally, and then we'll talk about the 3 diseases, well, really only 2 in detail. Sickle cell anemia, Tay Sachs, and then we'll broach and introduce the idea of cystic fibrosis, but of course the reading and presentation this week will fill in the many gaps for that disorder for us as well. So, first, just to talk about recessivity as a genetic principle, let's think about red hair. It's pretty incredible the length some people will go to have red hair. It shows how precious and valuable red hair is. No disrespect to Rihanna, but this is not red hair. Neither of these are red hair. This is red hair. Red hair is beautiful, especially when it's natural, and people want red hair because it is different. It stands out. It's unique. So the question for us as geneticists becomes, why is red hair unique? Why is red hair rare? What is it genetically about red hair that makes it different, makes it rare? And it all comes down to melanin, to natural biological pigment. In our cells, we have eumelanin, which is more of a brown and black pigment, and we have phosphomelananin, which is more of a red-yellow pigment. And the more overall melanin that one has, both kinds, the darker the hair, the darker the pigmentation, the less light. But more eumelanin gives browner hair if it's pumped into the hair follicle, and more phosphomelananin makes the hair redder. So that's all fine and good, but certainly that doesn't answer our question, which is, why is red hair rare? As geneticists, we're interested in understanding that why fewer people have the red hair phenotype, and what is it about their DNA, their genes, and their alleles that explains that. So this was found out quite a while ago through a mouse model. So scientists studying mice, and mice is a classic metazoan mammalian model organism for genetics. It's hard to work with genetically, but it's the closest thing we have that's easy to work with that tells us how our mammalian genetics work. But anyway, those scientists found a single gene that they named MC1R and found that it encoded a protein that allowed mice to have black or brown fur, depending on how it was mutated. This protein, when it's active in wild type, causes there to be more eumelanin and less phosphomelananin, and so the mice have a much darker, browner, blacker coat. But when this gene is mutated, you can probably figure out what happened. It flipped, so now there's less eumelanin and more phosphomelananin, and these mice were a bit ginger. Later, many years later, as the human genome was in the midst of being sequenced, other scientists found the same exact gene with a very, very similar sequence in our own genome. That gene in our genome lives on chromosome 16. And those human individuals that were screened for mutations in MC1R, those were red-haired individuals. So once again, this idea of comparative functional genomics, found the gene in ginger mice, found that gene then in us, and the gene in us does the same exact thing, makes us red-haired as well. So now we understand the genetic basis of red hair, but we still don't understand the rarity. Why is red hair rare? And so to do that, let's take a step back from the phenotype and think about genetics a little bit more holistically, a little bit more maybe mechanistically. Let's say that you own a factory, and your factory makes brown hair pigment. And on your factory floor, you've got 2 spots for 2 machines that make brown hair. These 2 machines are enzymes. Can you make brown hair if both of your machines work? If both of those enzymes are firing, you're churning out that eumelanin and you're making brown hair. So of course. Can you make brown hair if one of your machines work? Well, still the answer is yes. You're still capable of making that eumelanin. One enzyme is still working. You make plenty of that pigment, you have brown hair. But if both of those machines are broken, excuse me, then you have no enzyme to make eumelanin. The only melanin that can be made is phosphomelananin, and that's going to be a red-haired individual. So what we've done there is we've just equated classic Mendelian genetics with molecular genetics. Again, these machines are analogous to the enzymes, but what allows the cell to make these machines are the genes for this enzyme, or more importantly, the alleles from this enzyme. So if both alleles are mutant, then that proper enzyme can't be made, eumelanin can't be made, and you've got a red-haired individual. If either of the alleles is functional, however, and can give rise to a functional enzyme, then we can make this brown pigment and the hair will be brown. So we're beginning to scratch the surface of not only the Mendelian genetics and how it relates to molecular genetics and biochemistry, but we're also understanding the principle of recessivity as Mendel intended it. The reason why something is recessive is because it is less powerful than something that is dominant, i.e. a working allele, a working enzyme. So we know we get 2 alleles for each trait, one from mom, one for dad. We got different alleles on homologous chromosomes, and we also know that those alleles carry, or more specifically encode, the instructions for building a protein, in this case an enzyme that makes that eumelanin. If both of those genes are wild type, you show the dominant trait, brown hair. But the link between that genetic principle of being homozygous dominant and showing the dominant trait is with 2 dominant alleles, you've got 2 working alleles that encode 2 functional enzymes which give you the trait. If you've got one allele that's working and one that is not, you still show the dominant trait because just like our factory floor, you still are making the enzyme for eumelanin and you're still going to show that dominant phenotype, heterozygous. The way that you show a recessive phenotype, and the reason why recessive phenotypes are more rare, is because if neither allele is wild type, if both of those machines are broken and eumelanin cannot be made, only then do you show a different and unique phenotype like red hair. So we've got 4 possible outcomes here. 2 dominant alleles, 1 dominant and 1 recessive from mom and dad respectively, 1 recessive and 1 dominant from mom and dad respectively, or both recessive. And 3 out of those 4 outcomes gives you brown hair. Only 25% of them is red hair, and that's part of the reason why red hair is so rare. That's the molecular genetic reason why red hair is rare. Now there's a whole nother part of that story, population genetics and allele frequencies in the population, which we will not touch on in this course, but that also plays a role in rarity. So sometimes the recessive trait that we're studying is not cool and unique and enviable like ginger red hair. Sometimes that recessive trait is something debilitating, like sickle cell anemia, Tay Sachs, or cystic fibrosis. And that's what we're going to talk about mostly in this lecture. A few last points to consider before we do move on. Anything can be a phenotype, right? Red hair, eye color, sure, but also the predisposition for disease, including breast cancer. The ability for yeast to make the molecules that it needs to survive, such as the amino acid tryptophan or the nucleic acid adenine, that's a phenotype. The ability to make or not make a biological component. The cell's ability to repair its own DNA, yes or no, is a phenotype. And diseases are phenotypes as well. The presence of sickle cell, the presence of Tay Sachs, that's a phenotype. Also, it's important to note that for the most part, genes only get us so far. For most phenotypes, environment plays a role in that phenotype as well. Sometimes that role is enormous. Sometimes that role is exclusive. Most of the time, that role is intermediate. And this, of course, is the nature-nurture consideration. We can imagine a tree that has all of the alleles needed to be the tallest redwood in the forest. But if that seed for that tree lands in the shadiest, driest part of that glen, it's never going to grow tall. It's never going to absorb enough water or enough sunlight to make enough ATP to grow tall. So yeah, it might have had the alleles for tall redwood, but it's going to be a small 3-foot little shabby tree because it didn't have the right environment. And again, most phenotypes fall on this spectrum. Scurvy, right? That's a vitamin deficiency. That's entirely environmental. There's no genetic component to scurvy at all. Down syndrome, that's entirely genetic. There's really no environmental effect to having Down. But everything else in the middle, heart disease, diabetes, even hemophilia, these are on the spectrum between nature and nurture. Environment can play a role in the manifestation of these traits. So some traits, like the diseases that we'll discuss today, are primarily nature. And others are primarily nurture, such as scurvy. But always remember, alleles and genotypes is what we are passing on with DNA, not phenotypes. There is a huge genetic predisposition for addiction and alcoholism. Huge. But For that child of that alcoholic who grew up in that rough household and vowed to themselves to never have a drop of alcohol because of the destructiveness that they saw it observe, that kid, despite all those predispositions and all those alcoholism alleles, will never be an alcoholic because they can exert free will. That is the role of environment in a lot of traits. All right, so let's dive into sickle cell anemia. We'll start with an example that's familiar to many of us, an example that revolves around hemoglobin. Pretty sure we all know what hemoglobin does, at least in general, if we don't know the full mechanism. It carries oxygen in our red blood cells. So red blood cells move oxygen around in our bodies, carrying it from the lungs to the cells that need it, and hemoglobin is the compound in our red blood cells that does that oxygen carrying. Now here on the top we see a part of the DNA sequence of the normal, healthy, wild-type hemoglobin gene, and on the bottom we see the complementary sequence, well not complementary strand, just the version of the sequence from a mutant sickle cell anemia hemoglobin gene. And you can see the difference is very, very subtle. This is a base substitution, an adenine to thymine transversion. We've gone from a purine to a pyrimidine. So that's it. We switched an adenine to a thymine. And that one switch at that one base pair, and that's the only difference between these two alleles, results in red blood cells looking from what you see on the left to what you see on the right, sickle cell anemia. The red blood cells are highly distorted, and the symptoms of full-blown sickle cell anemia can be devastating. This one DNA sequence change, this one mutation, this one faulty base alters the hemoglobin protein enough that it can no longer efficiently carry oxygen. It distorts the shape of the red blood cells. Individuals with sickle cell anemia have debilitating joint cramps. They have blood vessel blockages. They have poor oxygen transmission, lethargy and fatigue. Really, really severe symptoms. And again, it's one base. So what's really going on here? How can the change of one single base have such a dramatic effect? And this is where we now are deeply into the convergence between molecular genetics and biochemistry. And we'll talk about how that DNA change results in a biochemical change, a protein level change in that hemoglobin protein. So that one incorrect base, that adenine to thymine transversion, it's called a missense mutation. Missense mutations in genetics is when there's an amino acid substitution in the protein that's made from the mutant gene. So in this case, the wild-type hemoglobin gene is supposed to have glutamate in that sixth position of the protein. And that one single base pair change results in a codon change, remember our genetic code, and now instead of glutamate or glutamic acid, we have a valine in that position. Now, structurally, valine and glutamate are pretty similar. If we look here, we see that they both start with the CH2 group. And then we've got, for glutamate, CH2, CH2, and then a carboxyl group. And here we have CH2, CH2, and CH2 and two methyl groups. Really similar in size, really similar in their carbon and hydrogen composition. I mean, quite similar all around, even in their topology, with that fork at the end. So, if they're so similar, why such a dramatic change in the shape of red blood cells, given this missense mutation, this single amino acid substitution? And it really comes down to their chemistry. Sure, they might be similar structurally and in terms of size, but glutamate is charged, right? Once we donate the proton off that hydroxyl group in glutamate, that oxygen there is negatively charged, full negative charge. And because it's negatively charged, glutamate is hydrophilic. It wants to be. It belongs on the outside of hemoglobin where it can favorably interact with water. After the mutation, we now have valine in that position. Valine is highly hydrophobic. Water shuns valine and pushes valine towards the center of the protein. So what we have is a complete distortion in the folding of hemoglobin because a hydrophobic amino acid is now on hemoglobin's exterior and water is pushing that valine deep into the protein and causing the protein to almost implode on itself and cause the protein to fold in on itself. This leads to a radically differently shaped hemoglobin, and that radically differently shaped hemoglobin distorts the red blood cell. In fact, when that valine cannot fold inward, the only other way to shield those valines from other valines is to aggregate hemoglobin. Now hemoglobin proteins stick to other hemoglobin proteins. All those exposed valines stick to one another, and by clustering together, they exclude water. just like droplets of oil in a vinaigrette dressing. But now you've got hemoglobin aggregating, and you make these big clumps of hemoglobin that distort the red blood cell. Very similar mechanism to Parkinson's and Alzheimer's in the brain. So this mutation is recessive. In order to have full-blown sickle cell anemia, an individual has to have 2 mutant alleles, one from mom and one from dad. However, given how debilitating this disease is, it's a fair question to say, like, shouldn't natural selection have taken care of this a long time ago? If it's so debilitating, if it leads to early fatality, then individuals with sickle cell anemia alleles shouldn't live to reproductive age. Why doesn't this kind of clear itself from our population? That question can be asked more crassly by just saying, why hasn't long ago everyone who's carrying these mutant alleles died due to a lack of fitness and took those alleles with them? That's how Darwinism works. We often say survival of the fittest. What we don't say about Darwinism is it also means death to the least fit, right? And by dying early, you take your faulty alleles with you. So the reason why that hasn't happened, and the reason why we continue to see sickle cell anemia now, is because there actually is a selective pressure for keeping this allele around. There's a benefit to the sickle cell anemia allele. What we have all the way on the left is normal red blood cells. This is a normal homozygous dominant genotype individual with completely normal red blood cells. And you'll see that such an individual is very, very susceptible to malarial infection. The malaria parasite can easily enter those red blood cells and infect them. All the way on the right, we see someone with full-blown sickle cell anemia. They've got 2 recessive alleles, so they're homozygous recessive, but these sickle red blood cells actually allow the individual to be resistant to malaria. Malaria has a hard time getting into these red blood cells, and so individuals who are homozygous recessive tend to not get malarial infections. That's not such a benefit to them because they're dying of sickle cell anemia, but they are incidentally resistant to malaria. The sweet spot in this case is the heterozygote. Individuals who are carriers for sickle cell anemia, so they have one wild-type allele, they've got a working factory machine, they're making working hemoglobin, they've got a fairly normal wild-type dominant phenotype, but they do have this broken valine-containing hemoglobin as well, and some of their red blood cells sickle as a result. These individuals have very, very mild symptoms of sickle cell anemia because so many of their red blood cells function properly. But conversely, they also happen to be quite resistant to malarial infections, and that's your fitness benefit. For a long, long time, as the human species was mostly in those tropical environments, malaria was a very significant pressure on our health and our survival. And any individual who was born with a natural resistance to malarial infection had a huge fitness advantage. They lived longer because they didn't get malaria, they mated more, and they got their alleles into the next generation. Turns out, though, a lot of those alleles were sickle cell anemia alleles, and hence the persistence in our environment still. So in other words, at least in this case, having one recessive allele is a good thing, but if you keep those recessive alleles around in those heterozygotes, you can't control how many homozygous kids are going to be had when 2 carriers mate. So because of this benefit, sickle cell anemia persisted and continues to persist in our species. So that opens the door to a really interesting discussion on Darwinistic natural selection in general, and especially to consider it from a genetics perspective or through a genetics lens. So natural selection is best described genetically as the differential reproduction of genotypes. Now sure, that's not what you're used to hearing, survival of the fittest, but that's really what natural selection is, at least for us geneticists. Again, differential reproductiveness, right? So some mating more than others, and that's due to genotype. We can kind of spin Darwinists' cliche of survival of the fittest and basically say what we mean here is reproduction of the fittest. If you're living longer, you're mating more, and if you're mating more, you're transmitting your alleles more efficiently to the next generation. The more offspring you have, the more of the next generation has your alleles. Beneficial traits, especially those that are genetically based, make it into the next generation more efficiently, and that's great because they're beneficial alleles. Those who live the longest mate the most because they're the healthiest and the most fit, so it makes a lot of sense that they put their alleles into the next generation. This usually skews allele frequencies towards better alleles. In other words, with each subsequent generation, you'll have a higher and higher percentage of beneficial alleles and a lower and lower percentage of harmful alleles. The reason being, those individuals in generation 1 with the harmful alleles died a lot younger, mated a lot less, and got fewer of those alleles into the next generation. And so we should march evolutionarily towards a greater and greater proportion of our gene pool being made up of good, healthy alleles. Now, this is great for species and great for evolution. Again, this is the mechanism by which Darwinistic selection moves forward. But the question then becomes, we return to, how would an allele, such as a recessive one for sickle cell anemia, persist in our human population? Well, we understand that from a health perspective, from the previous slide, we understand about the heterozygote, but what about through this lens of shifting allele frequencies? With natural selection and differential fitness just meaning some individuals are more fit than others, we can begin to predict what effects on allele frequencies we will see one generation to the next. And doing this is referred to as a general selection model. The results of this general selection model and the results of selection itself, when it does occur for a population, varies depending on which alleles give the most fitness. So here's a table that shows this kind of general selection model. This is probably one of the most important tables for evolutionary genetics that you can consider. And it's a little bit hard to navigate initially, but there's nothing here that you don't already know. So let me just orient you to the table, and then we'll go through some examples. We'll focus here on the type 1 fitness. So this W11, it's actually not W11. This represents a genotype, and it's a genotype where the two alleles are the same. So this really means homozygous. And the ones, the subscript ones here, refer to the dominant allele. So W11 really means two dominant alleles, homozygous dominant. Now, why they put the table in this W configuration is pretty much because evolutionary biologists don't use genetic notation, but they do give you a key down here where they say, no, W11 represents dominant-dominant. W12 represents dominant, and in this case, the 2 allele is recessive. So these are your heterozygotes. And W22 means 2 recessive alleles. These are your homozygote recessives. So homozygotic dominant, heterozygote, homozygotic recessive. What this is saying, then, is this equal sign, this greater than sign, the less than signs, all this refers to fitness. So we can decode this key as meaning the fitness of the homozygotic dominants and the heterozygotes is equal. And both of those are more fit than the homozygotic recessives. Well, that's Mendelism. Mendel basically told us that when we have genes exhibiting classic dominance and recessivity, there is no difference in phenotype between being homozygote dominant and heterozygote. And that's exactly what we're saying here. The fitness here is the phenotype, and there's an equal fitness between being homozygotic dominant and heterozygote, just like Mendel said. And that fitness is more or greater than being homozygotic recessive. Live longer, mate more, live longer, mate more, mate less, and die early, basically is what we're saying. So if these individuals here are living longer and mating more, it makes sense they're getting more and more of their alleles into the next generation. And the predominant allele here is the number 1 allele, the dominant allele. And with each subsequent generation, these poor individuals here, they're dying young and mating less, so with each generation we're getting less and less of the 2 allele, the recessive allele. And that's exactly our prediction. The dominant allele 1 increases with each subsequent generation, and the recessive allele 2 decreases, because the recessive allele here is less fit. So we can say that type 1 selection is telling us that the dominant allele confers fitness, and so does type 3 selection. Type 3 selection refers to a different class of genes called additive genes, but the general trend is the same. Here, the most fit is being homozygotic dominant. Dominant alleles are good. They make you live longer, they make you stronger, they make you more fit, and you mate more. Here, being homozygotic recessive is the least fit, just like in type 1. Recessive alleles are bad, unhealthy, you die young, you mate less. What's different between type 1 and type 3 is that the intermediate phenotype now has an intermediate fitness. In other words, the heterozygote has its own unique phenotype of fitness. They are a little bit more fit than the homozygotic recessives, but they are less fit than the homozygotic dominants. Doesn't matter really, though, for an allele frequency point of view, because still, the more dominant alleles you have, the better off you are. And so with each subsequent generation, we're going to have more and more dominant alleles in the gene pool and less recessive alleles. Type 2 and 4 natural selection are also directional. Directional selection means with each subsequent generation, we're having more and more of one allele and less of another. So again, with each subsequent generation, the frequency or proportion of the dominant A allele will increase and the frequency of the recessive A allele will decrease. Directional selection. And as I said a moment ago, type 2 and type 4 are also directional. We're seeing allele frequencies change one generation to another. So how are 2 and 4 different than 1 and 3? Well, again, if these mathematical symbols mean fitness, in type 2 directional selection, the most fit phenotype, or genotype, is being homozygotic recessive. But we are abiding by Mendel's laws. Mendel always tells us that being homozygotic dominant is equal, phenotypically, to being heterozygotic. Being homozygotic dominant is equal, phenotypically, to being heterozygotic. We're staying true to Mendelian principles here. It's just that the dominant phenotype here is being less fit, and the recessive phenotype is being more fit. So when it comes to living longer, mating more, and getting your alleles into the next generation, the winner in type 2 natural selection are the homozygotic recessives. And so with each subsequent generation, the proportion or frequency of the recessive allele goes up. And having dominant alleles makes you die young, so the proportion of dominant alleles goes down. It's directional, but it's directional in favor of the recessive allele. And type 4 follows the same pattern, it's just that type 4 is additive, not Mendelian. So instead of the homozygotic dominant and heterozygote having an equal fitness, we have an intermediate phenotype. Still, being homozygotic recessive is the most fit in type 4, but being heterozygotic is intermediately fit, and being homozygotic recessive is least fit. Homozygotic dominant is least fit. So despite this intermediate pattern and this additivity, if you want to live long, if you want to get your alleles into the next generation, in type 4, you definitely want to have recessive alleles, and so the proportion of recessive alleles in the next generation will go up, and the proportion of dominant alleles will go down. Now, some of you are probably wondering at this point, well, aren't these mutually exclusive? How could you be type 4 and type 1? And you're absolutely right, they are mutually exclusive. You can't be both. Different genes follow different patterns of natural selection. So we're going to talk in class with a challenge question about which one of these types sickle cell anemia falls into, but different genes, different disorders, different phenotypes follow one of these models. So one gene might be type 2, and another gene might be type 4, a third gene might be type 1, but it is impossible for a single gene to be more than one type of natural selection model. So again, types 2 and 4 also directional. They're just favoring the opposite allele, the recessive allele. Eventually, directional selection, if left unchecked, will lead to what we refer to as extinction of the lesser, least fit allele. When that happens, we don't have any more of those alleles in the population, and 100% of the gene pool is made up by the more fit allele. This is good. This is Darwinism from a genetics perspective. We want to clear out the recessive alleles and only keep the helpful alleles, the fit alleles. Now, types 4 and 5 are unique in that the heterozygote has its own individual fitness and it's not additive. Type 5 general selection is called overdominance, and it's defined by the heterozygote actually being the most fit and being more fit than either homozygotic recessive or homozygotic dominant. In the case of overdominance, we actually reach a stable equilibrium where both main alleles are maintained in the population into perpetuity, actually, unless something skews that balance. So there's never allele extinction. The reason why is because the most fit is the heterozygote, and the only way you can be heterozygote is if you've got a dominant and a recessive allele. So in order to make the most fit genotype, you need both alleles in the population, and that means the population is being selected to maintain both alleles. So this might look familiar, and like I said, we'll talk more about it on Friday in class. And I should say type 6 is really way out there. In type 6, the least fit genotype is the heterozygote. The homozygotic dominant and homozygotic recessives are more fit, but differentially, and can't make any sense of underdominance. It's very unstable. The allele frequencies change almost randomly, almost like a statistical noise one generation to the other. It's a population geneticist's worst nightmare, underdominance. So now let's move from sickle cell anemia to Tay Sachs disease. Tay Sachs disease is also a genetic and recessive disorder. Again, that's the theme of this lecture. It's caused by a mutant gene on chromosome 15, and that mutation results in an enzyme that is non-functional. The enzyme that has been struck down by this mutation is, and I'm not good at enzyme names, but hexosaminidase A is going to be my best shot on that one. And when that enzyme cannot work properly, we see a buildup of fatty deposits in the cell as well as cellular waste. And this mutation and this syndrome specifically affects neurons the greatest. So again, if we look at this schematically, we've got this gene mutation on chromosome 15. We can't make this hexa enzyme as a result of that mutation. And we've got these fatty molecules, these gangliosides that are building up in this neuron. Hexa should be breaking them down into substituent parts that can be recycled, but without Hexa, these fatty acid deposits accumulate in the lysosome of the neuron, and the neuron's health begins to deteriorate as a result. In fact, the neuron will ultimately die because of faulty lysosomes and the buildup of these fatty deposits. Somewhere between the onset of those buildups and death, we see cognitive development defects because of those neurological cells, those neurons not working properly. So we can see it a little bit better in this schematic. Here's a healthy neuron, and here's a Tay Sachs neuron. The lysosomes, small membrane-bound organelles that are the, quote-unquote stomach of the cells. I'm sure somebody has said that to you before. There are digestive enzymes in the lysosomes that not only will digest nutrient molecules that are sometimes phagocytized from the outside environment, but a lot of the recycling of cellular components occurs in the lysosome. Proteins are broken down, fats are broken down into their constituent subunits, and then those blocks are reused and recycled by the cell to make new complex molecules. So the lysosome is really the recycling center of the cell rather than the stomach of the cell, in my opinion. So in this Tay Sachs cell with the mutation, we see that the lysosomes are bulging. They're bulging under the pressure of unmetabolizable recycling products. It's almost as though the recycling center has broken down, but the trucks are still delivering the plastic and the glass. And so the lysosomes are bulging under the pressure of the waste they continue to accumulate, but are unable to process. And ultimately, that's going to cause destruction to the cell. The cell is going to break down because of these ineffective lysosomes. So 5 to 6 months after birth, those with Tay Sachs start to develop blindness, paralysis, and other acute neurological symptoms. And sadly, death usually occurs by age 6. Now, here, the reason why Tay Sachs is still a disorder that is quite prevalent and common, at least in some populations, is not because of the heterozygote having any benefit. Right? It's not the same story as sickle cell anemia, where persistence is due to there being a desire to keep the recessive allele. Instead, the persistence of the disorder here is due to non-random mating. One of the key defenses we have to get those alleles out of our population is primarily random mating, going outside of our family, of course, outside of our group, outside of our tribe, and diversifying our genome, diversifying our allele composition. When we don't do that, we start to see inbreeding effects. We start to see the accumulation of recessive alleles because our tribe, our group that we are breeding with, is not representative of the allele frequencies of the overall population. So globally, the prevalence of Tay Sachs is 1 in 35,000 live births. It's a rare disorder, a very, very, very small percentage. But specifically, in Jews of Eastern Europe descent, that prevalence is tenfold higher. It's 1 in 3,000. And shockingly, in a specific sect of those of the Jewish faith called Ashkenazi Jews, we see 1 in 27 individuals are a carrier. They're heterozygote for Tay Sachs. Now remember, Tay Sachs is lethal, so no one lives to adulthood with it. 1 in 27 individuals are carriers for Tay Sachs. That's a large amount. We're talking about 3-4% of individuals are carrying this recessive trait, and they're carriers. So we know what that means, right? We can imagine the cross. If two individuals, a man and a woman, mate, and they're both carriers for Tay Sachs disease, they've got a 25% chance of having a child who has Tay Sachs. That child will die usually by the age of 6. But what we sometimes don't appreciate is they also have a 50% chance of having a child who's a carrier. And this carrier child will grow up, have their own kids, and continue to propagate this recessive allele. So it's the non-random mating among this sect of those of this faith that are keeping this allele in the population. There's lots of things that go into allele balance. And sure, fitness is a big part of that. But so is no migrating, so is random mating, so is very large populations. And the more that we diminish those characteristics and have different fitness profiles, we begin to migrate, we mate within small populations non-randomly, the more we don't get that outcrossing effect. We continue to maintain these deleterious alleles, and mutations and disease persist. So finally, for cystic fibrosis, or CF. Cystic fibrosis is caused by a mutant allele on chromosome 7, also recessive, that's why we're talking about it here. This disorder is caused by a defective membrane protein. More specifically, it's a chloride transporter, a membrane transporter for chloride atoms. And because this protein is actually mildly misfolded, the truth is, if this mutant chloride transporter were able to get into the cell membrane, it would actually function. But it's slightly mutant in its folding, and that misfolding traps it in the ER, the endoplasmic reticulum. So this mutant chloride transporter just can't get to the cell membrane because it's kept in the ER. The lack of that chloride transporter at the cell leads to mucus buildup in the lung, in the airways of the lung. The balance between sodium and chloride is a huge part of how we regulate water flow in our cells physiologically. And with chloride not being able to be moved, water doesn't move properly, and water actually accumulates in the airways of the lung and leads to mucus buildup. And that watery, wet, mucousy environment of the lungs is actually the perfect fertile breeding ground for bacteria. Bacteria can get trapped in that liquid. Life needs water to survive, but they can feed on the proteins that are there as well. And so cystic fibrosis is characterized by really wicked lung infections, but it's due because of where chloride isn't going, interestingly enough. So this buildup of fluids in the lungs creates this hospitable environment for bacterial infections, and that's the real danger and the real disorder of cystic fibrosis, that infection. So why does this allele persist? Right? This has to be something different. Surely there's no benefit to having wet, bacterially accommodating lungs, and I'm not aware of any tribalism for cystic fibrosis. So why does this mutant allele continue to persist in our environment? And it's so interesting because if you dig deep enough, everything makes sense. Not all that long ago, about 12 years ago, this paper was published where it was identified that cystic fibrosis recessive allele, when present in heterozygotes, just like sickle cell anemia, seems to provide a protection against tuberculosis, another really wicked respiratory infection that hit our species very, very hard over time. So Darwinism, really, really good at getting rid of deleterious low fitness alleles, right? Natural selection does an amazing job of cleaning out our genome and promoting evolution. Those harmful alleles, if they're still around, there's always a reason why. Non-random mating, fitness advantage of the heterozygote. You just got to dig deep enough. And so, with our introduction to this last unit almost complete here, we begin to see that not only can non-Mendelian principles be explained if we just look deeply enough, and that's what we try to do in Unit 1, but also, interestingly, non-Darwinian phenomena can be explained as well by digging just as deeply and looking at these things through a Darwinistic and genetic lens. So, to summarize what we try to talk about today, you get 2 alleles for each trait. right? You get one from your mother and one from your father. We're all diploid, and we are all composed of homologous chromosomes. Those alleles encode instructions for building proteins. That's the central dogma. But genes only get you so far. The environment plays a huge role in a lot of phenotypes as well, and that's that nature versus nurture consideration. Then, with that molecular genetics in our back pocket, we dove into the disorders. In sickle cell anemia, we've got one incorrect base, that adenine to thymine base substitution, and that leads to a missense mutation. Glutamate becomes valine, an amino acid substitution. Hydrophilic becomes hydrophobic, and the proteins don't behave properly. This mutation, even though it's recessive and requires 2 mutant alleles to have anemia, is around still because of the selective pressure for the heterozygote. Heterozygotes of sickle cell anemia are resistant to malarial infection, and so keeping those recessive alleles around was a good thing for a very long time in our species, and continues to be a good thing in a lot of tropical environments. That led us to consider the general selection model, where we have type 1 and type 3 favoring the dominant allele, type 2 and type 4 favoring the recessive allele, and type 5, over dominance, favoring the heterozygote. We went on to talk about Tay Sachs disease, also a recessive disorder, resulting in cognitive defects developmentally, and we learned a little bit about the physiology and the biochemistry of that with hexa and the buildup of bulging non-functional lysosomes. And we closed out just scratching the surface of cystic fibrosis, teeing it up for the group that's presenting this week, a recessive disorder that leads to wet lungs, hospitable for bacterial infections, but when we dig deep enough, we see that type 5 overdominance seems to be playing a role there as well, where the heterozygote actually has a fitness advantage. So hopefully you find this material to be as interesting as I do and enjoyed our first foray into the last unit, human disorders, and I'm so looking forward to your questions, what you'd like to talk more about on Friday. We'll hit some challenge questions really hard, interesting challenge questions in this unit, and then can't wait to hear more about cystic fibrosis as well. Until then, thanks for watching, and have a great day.